Hello students, welcome to my channel. So in today's lecture, I am going to discuss on the topic on classification of carbohydrates. Alright, so today we are going to discuss on this topic. Now, carbohydrate molecules, they are classified into four different classes, okay, into four main classes, that is the monosaccharides, then we have the second class, that is disaccharides, the oligosaccharides, and the polysaccharides. So these are the four different classes of carbohydrate molecules, and in today's lecture, I am going to discuss only on the first class of carbohydrate molecules, that is monosaccharides. For these disaccharides, oligosaccharides, and polysaccharides, I'm going to make a separate video for these. So today we are going to let us discuss on the first class of carbohydrate molecules, that is the monosaccharides. Now, what are monosaccharides? The monosaccharides, they are the simplest group of carbohydrate molecules. They are also often called as the simple sugars. Okay, simple sugar molecules, since they cannot be further hydrolyzed. Okay, they cannot be broken down any further. They are the simplest sugar molecules that consists of only one unit of simple sugar. Only one unit of simple sugar. So these are the simplest group of carbohydrate molecules. Now, these monosaccharides, they are colorless, and they are also crystalline solid, which are soluble in water and insoluble in they are soluble in water and they are insoluble in non-polar solvent. So these monosaccharides, they are those saccharide molecules, they are those sugars that can be absorbed and utilized by the body. Okay, they are those simplest monosaccharide sugars, carbohydrate molecules that can be absorbed and utilized by the body. So here the monosaccharide molecules, this class of carbohydrates, okay, now there are three monosaccharides that are particularly important in the study of nutritional science, and those three important monosaccharides, they are the glucose, then we have fructose, and we have galactose. So today we are going to talk about these three in particular, okay, one by one. Now, in monosaccharides, now these, just remember that these are the three important, particularly important in the study of nutritional science, these three monosaccharides. Now in monosaccharide molecules, the number of atoms, okay, in monosaccharides, the number of carbon atoms, because we know that for that I had prepared a separate video on the carbohydrate molecules. So we know that carbohydrate molecules, they are made up of carbon, hydrogen and oxygen, right? So for the detailed explanation of this, you can watch my other video, particularly on this topic, on the structure of these of the carbohydrates. Now in monosaccharides, just remember that the number of these carbon atoms, it usually ranges from 3 to 7. Okay, it usually ranges from 3 to 7. Now these monosaccharides, they can be classified on the basis of the number of carbon atoms that are present in the molecule, and those classification are diose, then we have triose, then we have tetros, then we have pentoses, hexoses and heptoses. Okay, so depending upon the number of carbon atoms which are present in a sugar molecule in a monosaccharide, they have been classified into dioses, trioses, tetroses, pentoses, hexoses and heptoses. That means dioses, they have 2 carbon atoms. For example, one of the example, which the monosaccharide molecule which has only 2 carbon atoms is glycolic aldehyde. Alright, glycolic aldehyde. Now triosis meaning 3 carbon atoms, and the one of the best example of triosis monosaccharide molecules is glyceraldehyde. Then we have the tetrosis, that is, which consists of 4 carbon atoms, and the example is erythrose. Okay, then we have 5 carbon atoms, that is pentoses. Example of this is ribulose. Then we have hexoses. The very common example is glucose and galactose. And we have heptoses, that is, which contain 3 carbon atoms, and though the example of heptulose is pseudoheptoses. So the monosaccharide, they can be classified on the basis of the number of carbon atoms which are present in molecule into these different classes. Okay, because as I said, that the number of carbon atoms ranges from 2 to 7. Okay, so but there is another type of classification. The monosaccharide molecules, they can also be classified on the basis of the functional groups. Alright, on the basis of the functional groups, that is according to the nature of the reducing or the functional groups present, namely aldoses and ketoses. So what are aldoses? Aldoses are those in which the reducing group is the aldehyde group. Alright, the aldehyde group. And ketoses are those in which the reducing group or the functional group is a ketone group. So the monosaccharides, they can also be classified into aldoses or ketoses depending on a functional group. If it's aldehyde, then those are classified as aldoses, and if the functional group is a ketone, then those are classified as ketoses. So the presence of these functional groups in a molecule, it results in the reducing property of the monosaccharides, all right? And this reducing action, it can be determined by Felling's or Benedict solution, where these sugar molecules, these monosaccharides, they convert the cupric hydroxide molecule to cuprous oxide. Okay, therefore these type of monosaccharides, they are, or these type of sugars, they are called as reducing sugars. So monosaccharides, they are therefore called as their reducing sugars. And now, okay, I hope that is clear that they are classified based on the number of carbon atoms present, and also based on the number of based on the functional group present, whether it is an aldehyde or a ketone. Now, the 3 carbon monosaccharides are known as, we know that the 3 carbon monosaccharides are known as trioses, right? Those which have 3 carbon atoms in their molecule. So for this example is glyceraldehyde and dihydroxyacetone. These two are the important trioses in the biological system. Alright, they are the important trioses in the biological system. And here the structure of these two, I can show the structure of a simple aldose and a simple ketose, because one of this one is an aldose and this one is a ketose. So let us take a look at the structure of glyceraldehyde. So this is the structure, the straight chain structure of the linear chain structure of glyceraldehyde, okay, which is an aldose. Why it is an aldose? Because it contains the aldehyde group, aldehyde functional group, whereas the dihydroxyacetone, its structure is this is dihydroxyacetone, and based on the functional group here which is present, since it is a ketone, then this is referred to as a ketose, okay. And according to the number of carbon atoms present, then both these monosaccharide, both these sugar molecules, they fall on the trioses. Okay, they are the trioses, meaning they have 3 carbon atoms in their molecule. So the most monosaccharides, their names, they usually end with the suffix ose, ose. Okay, for example, you will see many monosaccharide molecules. For example, here glucose, galactose, ribose, lactose, then fructose, etc. All these, their names, they are ending with the suffix O's. Okay, and the one of the most abundant monosaccharide in nature is the 6-carbon sugar called the glucose. Okay, so glucose is a 6-carbon sugar, meaning it is a hexose. All right, having a 6 number of carbon atoms in its molecule. Now the glucose molecule, okay, the formula of glucose molecule is C6H12O6. Now this glucose molecule is the most important monosaccharide in metabolism. Why? Because it is the starting substrate of respiration to extract the energy which the living organisms needs for its maintenance. And during cellular respiration, what happened, the energy is released from glucose, and that energy is used to help make adenosine triphosphate. So glucose is the main source of energy. Now in the plant world, glucose is widely distributed. In case of plants, it is found in all plants and in the phloem, the sap of trees, fruits, vegetables are all wholesome food sources of glucose. And it is also present in other substances such as molasses, honey, corn syrup. All right. So plants, how they synthesize glucose? They are synthesizing glucose using carbon dioxide and water. And glucose in turn, which are synthesized by the plants, in turn is used for the energy requirements of the plants. And after the plant synthesize glucose, the excess glucose is often stored in the plants as starch. Okay, that is catabolized, meaning that it is broken down of larger molecules by the cells. By humans and other animals that feed on plants, they will break down starch in order to obtain glucose, all right, to use as energy sources. Now how this glucose is formed from the hydrolysis of di and polysaccharides. So disaccharides, those saccharides which contain 2 monosaccharide units linked together, and polysaccharides, poly means many, those carbohydrate molecules which are made up of many monosaccharide units. All right, so the breakdown of these di and polysaccharides, the hydrolysis of these saccharides, we can obtain from the hydrolysis of these saccharides, we can obtain glucose molecule. So glucose can be obtained from the hydrolysis of starch, dextrin, maltose, sucrose and lactose. Alright, that means from the hydrolysis of di and polysaccharide molecules. Now before we talk about the structure of glucose molecule, okay, before we are going to discuss on the structure of glucose, now let us first take a look at the structural representation of monosaccharides. Okay, the structure of monosaccharide molecules. Now the first one, that is the Fischer projection. Okay, the Fischer projection is actually the representation of monosaccharide molecules in linear form. Alright, in the linear form. Now there are several ways to draw the structure of a monosaccharide, of a carbohydrate molecule, and the Fischer projection, that is the straight chain, alright, straight chain representation, it makes the molecule appear flat. Alright, For example, here I have shown the structure of a glucose molecule. So the Fischer projection, it makes the molecule to appear flat, but it is actually a three-dimensional molecule. It is a three-dimensional molecule. And the Fischer projection, it does provide the information about the orientation of the atoms around each carbon atom. Okay, and this Fischer projection, that is this representation of monosaccharide, was devised by a German chemist. His name is Emil Fischer, in the year 1891. Okay, so this linear form or straight chain representation of monosaccharides was devised by Emil Fischer in the year 1891. And in a Fischer projection, the carbohydrate is shown in its open chain form. Alright, as you can see here, the glucose molecule, I am showing you here the glucose molecule. So it is shown in its open chain form rather than a cyclical one, because we will study later the cyclic representation of carbohydrate molecule. Now, carbon atoms in the main chain, you can see here these carbon atoms in the main chain, they are connected vertically here. They are connected vertically, while the hydrogen atoms and the hydroxyl groups, they are bonded horizontally. So this is referred to as the Fischer projection representation of monosaccharide molecules. Okay, the Fischer projection. Now the horizontal lines, they illustrate the bonds which come out of the page this way. Alright, whereas these vertical lines, they show the bonds that are in the page. Because like I said, this molecule here which I had shown, the glucose molecule, if you take a look at the straight open chain form of the glucose molecule, the Fischer projection, it looks flat, the molecule, but it is actually a three-dimensional molecule. So just remember that these vertical lines or these horizontal lines, they illustrate the bonds which come out of the page, whereas these vertical lines, they show the bonds that are in the page. And the carbon atoms may or may not be shown in case of Fischer projection. Sometimes you will suppose in case of a triose molecule, sometimes you will find like this. Suppose okay, you can see here that the carbon atoms are not shown. So it depends. Sometimes the carbon atoms may be shown or may not be shown in case of Fischer projection. And Fischer projection, it has one main advantage. It is easy to visualize and identify the stereochemical properties of a carbohydrate. And if you don't know about the stereochemistry of carbohydrates of monosaccharides, that also I have prepared a separate video. You can watch it on my channel. I will upload that on my channel. So when we talk about the stereochemical properties of the carbohydrate, it is easy to compare, to visualize, or to identify using the Fischer projection, to identify quickly and easily. For example, if I have two enantiomers, alright, if I have two enantiomers, that means the molecules which are mirror image of each other, it is easy to identify between those two enantiomers. Alright, the two enantiomeric molecule, it is easy to identify between those two using the Fischer projection. Now, to take the example of the glyceraldehyde molecule, as you can see here, okay, the mirror image of this one, alright, so it is easy to identify the enantiomers using the Fischer projection. It is easy to identify between these two enantiomers of glyceraldehyde, okay, which are mirror images with each other. As you can see here, this is the structure of a glyceraldehyde and they are the enantiomers. You can see it is easy to identify using the linear open chain structure of glyceraldehyde using this Fischer projection. So here, as you can see, a Fischer projection, it provides an easy way to distinguish among many similar carbohydrates, okay, those carbohydrate molecules or monosaccharides that have similar structure. Now, for example, we know that there are 16 aldohexoses. Aldohexoses are those molecules. Why aldo? Because they have an aldehyde group. As you can see here, example, I have given 4 of the 16 aldohexoses. Aldo because they contain an aldehyde functional group, and hexoses because they have 6 number of carbon atoms in their molecule. So these are the example of the 4 of the 16 aldohexoses that are similar in structure. Now, note the difference of the Fischer projection for each of these molecules. So, changing the orientation of one or more of the hydroxyl groups, it changes the identity of the molecule. If you see the glucose, this molecule here, the glucose, as you can see, what is the difference between the glucose and the allose is at the carbon, here it should be OH and here it's H. So by changing the orientation of the hydrogen and the hydroxyl bonds here, the OH bonds on the left and right side of the Fischer projection, that means in carbon number 2, now it gives a different carbohydrate molecule. Okay, we get a different monosaccharide molecule, that is D-allose. So the difference between these two is only at carbon number 3 here. 4, 5 and 6. And just take a look at the structure of glucose and mannose here. What is the difference is in carbon number 2, where the hydroxyl group in case of glucose is on the right, whereas in mannose is on the left. And the same thing with the galactose, the difference here is at the carbon number 4, as you can see. The carbon number 4, as you can see, the hydrogen is on the right, the hydroxyl is on the left, whereas in glucose the hydroxyl group is on the right. So that is why the Fischer projection, it provides an easy way to distinguish among these similar carbohydrate molecules that exist in nature, and thus changing the orientation of just one or more of the hydroxyl group, it changes the identity of the molecule. Now, the next structural representation of carbohydrate molecules is Howard projection. Now, most of the monosaccharide molecules with 5 or more carbon atoms in backbone, they usually occur in aqueous solution as cyclic or ring structure, all right, and not in straight chain and not in Fischer projection. So a Howard projection, it differs from a Fischer projection in that it is used to represent a carbohydrate molecule in its cyclical form and not in an open straight chain. Now this ring structure, it was proposed by Howard in the year 1929. And he based his proposal on the scheme in which all the sugars forming either 5 or 6 membered rings, they are termed as furanoses. These 5 membered are termed as furanoses and the 6 membered as pyranoses. Okay, so the 5 membered rings of monosaccharide molecules, they are termed as furanoses, whereas the 6 membered, they are termed as the pyranoses. So the Howard projection, they represent the carbohydrate molecule in its cyclical form. Here the pyranose ring of the glucose, the pyranose is the 6-membered cyclic hemiacetal which is formed from aldohexose. Okay, these aldohexose, they usually form a 6-membered ring termed as a pyranose ring, which this name, it comes from a 6-membered cyclic ether, that is pyran, and furanose is the 5-membered cyclic hemiketal formed by the ketohexoses fructose, and the name, it comes from a 5-membered cyclic ether named furan. Now pyranose and furanose rings, they can be represented by Howard projections as it is shown here, and these Howard projections, they are planar representations of furanose and pyranose forms of carbohydrates. Now these types of projections, as in this example, they allow the cis-trans relationship among the hydroxyl groups to be seen, in which the ring is drawn as if it was it were flat and is viewed etched on with the oxygen atom, as in this case of furanose, with the oxygen atom at the top in case of furanose, but in case of pyranose, you can see here the oxygen atom is at the upper right of the ring. And the relationship between the Fischer projection and the Howard projection, it's very important to remember that the group on the right, these are on the right. Okay, the group which are present on the right in case of a Fischer projection is drawn down in case of Howard projection. For example, just take a look at this. You can see these on the right, they are below this ring. Okay, they are down the ring, whereas the group on the left in the Fischer projection is up in case of Howard projection. You can see it is up here, right? This, you can see the OH, this one H and this one. So just remember, when you draw the Howard projection, okay, when you give the Howard representation, the Howard projection representation for any monosaccharide, that the molecules, the atoms, the groups which are present on the right should be down and on the left should be up. And for D sugars, for example, here in case of D sugars, this terminal CH2OH group is always up in case of Howard projection. You can see here the carbon number 1, 2, 3, 4, 5, 6. So just remember this last group here, the CH2OH group, the alcohol group, is always drawn up in case of Howard projection, and the ring structure is numbered in clockwise direction, all right, in this direction, as you can see here, next to the oxygen. So next to the oxygen, we have to number the carbon atom. You can see 1, 2, 3, 4, 5, 6. So just remember these rules when you give the Howard representation for any carbohydrate molecule. So for D sugars, the OH group, now this is the example of a D fructose, a D sugar molecule, that is a D fructose. The OH group at the anomeric position, here also the anomers, if you don't know, for those who don't know or for those who want the explanation of this, I had given in a separate video. I had explained about these anomers. Now we have two anomeric form of a carbohydrate molecule, alpha and beta, right? So how you will identify, in this case you see this is an alpha anomer and this is beta anomer. So how we will identify that? You just take a look at the anomeric carbon atom. Here this one is the anomeric carbon atom, and in this case this is the anomeric carbon atom. Now if you take a look at the anomeric carbon atom, the anomeric position, the position of the OH group or the hydroxyl group, in case of alpha anomer, it is drawn down. That is, it is opposite to the CH2OH group, because the CH2OH group here is always up. So if the OH group is down, is opposite, the position of OH group is opposite to the alcohol group, then this anomer is referred to as the alpha anomer. But if it is on the same side, as in this case, you can see that the OH group in the anomeric carbon position, carbon atom, the position of OH group is up, and it is on the same side as compared to the alcohol group, the CH2OH group. So in this case, this anomer is referred to as the beta anomer. So those are the important points to be noted, the important points to remember. Now for this is in the case of the sugar molecule, the D sugar molecule, whereas for L sugar, all right, now for L sugar, because we will see that later, I will show the L sugar molecule, whereas for L sugar molecule it will be the opposite. If the OH group, it lies, if the OH groups at the anomeric carbon is down, then it will be alpha, and if it's up, it will be alpha, and if it's down, it will be beta. Why? Because for L sugar molecule, this CH2OH, it will always be here down. Now CH2OH is here. This is the last carbon atom, meaning the last carbon atom is down in case of L sugars. So this also will change now. Why? Suppose in this case you see that the OH group is on the same side of CH2OH, then it will be beta and not alpha. and if it is on the opposite side like this, then it will be alpha. So the rule to remember is that just remember that if they are on the same side, that is the hydroxyl group and the alcohol group, they are on the same side, then they will be beta, and if they are on the opposite side like this, they will be called alpha anomer. All right, so this is the case of the L sugar because the CH2OH is down, but as in the case of D sugar, as in this case here, in this example, it is a D sugar, so the CH2, the last carbon atom, that is the last carbon atom in this group, the CH2OH group, is always on top. It is always drawn up in the ring structure. So in this case, then this will be alpha because they are opposite. This case it will be alpha, and in this case it will be beta. Why? Because here they are on the same side, it will be beta, and opposite alpha. So it is very important to remember that when you give the Howard projection representation of monosaccharide, just remember that alpha, they are opposite side, and beta same side. Okay, the hydroxyl group and the CH2OH group, they are on the opposite side, whereas in case of beta anomer, the hydroxyl group and the CH2OH group, they are on the same side. I hope it's clear. Okay, so here let us do this together. All right, for this is the example of hexose sugar, that is D-glucose. Okay, this is the D-glucose, the D sugar, and it is in linear form. Now let us draw the Howard projection of the D-glucose molecule. Now as a rule, we have to these structures on the right, these structures on the right, they should be down. Okay, starting from the first molecule. So here it will be here you have to number from the oxygen atom. We have to start numbering the carbon atom in a clockwise direction. So this will be the first carbon, second, third, fourth, fifth, and sixth. Here is this CH2OH, the sixth carbon atom. Because it is a D-glucose molecule, so the last carbon atom should always be up. Alright, so here also it will be CH2OH. Now this is carbon number 1, 2, 3, 4, 5, and 6. Now if you want to draw these groups on the right, they should be down. So here it will be OH, then we have here H, then OH. You can see this should be down, OH, then H, then this one, then here also OH, right? And here we have in the last carbon H, and these on the left you should draw up. So starting from this carbon number 1, that is H, then we have here OH, then this one H, this one also H. So you see how to draw these. On the right should be down and on the left should be up. And in case of D-glucose, the alcohol group should be up. So this is the Howard projection. So the carbon number, here we see the carbon number 1, it is referred to as D, anomeric carbon. Alright, now you have to take a look at the anomeric carbon. Now sometimes when we know that there are two anomers, the alpha and the beta anomer. In case of the alpha anomer, we know that the hydroxyl group on the anomeric carbon number 1, that is carbon number 1 here, it lies in the opposite side as compared to the CH2OH group. So that is the alpha carbon. But in case of beta anomer, the hydroxyl group here in the anomeric carbon, it usually lies up, okay, in the same direction as the CH2OH group, but the rest will be the same. Okay, those groups which are on the right side, they will be down. Alright, so here it will be the same, H, then this OH, again OH, here we have H, then these will be above. But just take a look at the anomeric carbon atom. Here the hydroxyl group, it lies on the same side of the CH2OH. So this is the beta form or the beta anomer of glucose. And this is referred to as the Howard representation, that is a 6-membered ring structure pyranose. Okay. Coming to the now coming to the chair and boat configuration, okay, of monosaccharide molecules. Now the 6-membered ring structure, that is the pyranose ring of a 6-carbon aldose sugar, okay, of a 6-carbon aldose sugar, it can exist in either a boat or a chair conformation. A boat on a chair conformation, and as you can see here that this figure A, it is the boat conformation, and figure B, it is the chair conformation of a pyranose sugar. Alright, a 6-membered carbon aldose sugar, where A here, it means the axial bond and E means the equatorial bond. Now from the figure, what do we see? We see that the substituents attached to the ring carbon that extend parallel, you can see here, these that extends parallel, they are referred to as the axial. They are said to be axial, that is represented by small letter A. Whereas those that extend outward from the axis of symmetry, now this is the axis of symmetry, alright, so those that extends away or outward from the axis of symmetry, they are referred to as the equatorial, represented by a small letter E. And in the boat form, actually there is a considerable steric hindrance between these various groups attached, okay, to the ring, to the carbon atom of the ring, and therefore this boat form, it is less favorable energetically. Hence the chair form, it predominates, okay, as you can see here in figure C. In the case of a beta-D glucose molecule, in which all these axial positions, they are occupied by the hydrogen atoms. Okay, so this is another representation of monosaccharide molecules, the structural representation of monosaccharide molecules, as in this example you can see of a beta-D glucose molecules, which is a very stable chair conformation. Okay, so coming back to the structure of a glucose molecule, which is a very common example of monosaccharide molecules, most important monosaccharide, alright, the most important and also the most abundant monosaccharide. Coming back to its structure, as we have already seen that the D-glucose, this is in the linear form or the open chain structure of the glucose, and as you can see here, this is the Howard representation of D-glucose in a pyranose ring, which form a pyranose ring. Okay, it is the Howard representation. Now, what happened when an aldehyde group, this one, the carbon number 1, the aldehyde group at carbon number 1, it react with the hydroxyl group at carbon number 5. Alright, so if it reacts with the the hydroxyl group on carbon number 5, then cyclic hemiacetal is formed. All these I had explained in a different video, so you can watch that video so that you will understand. Now, hemiacetal is two types of cyclic hemiacetal are formed, that is alpha and beta. Here beta, because the OH and the CH2OH are on the same side, so it is a beta form, and this is an alpha form. And how you will name this is also named as beta D, since it is a D sugar, this one, because we have the OH group on the right. So since it is a D sugar, then this can be named as beta D glucopyranose. Okay, because it forms the 6-membered ring, so it is referred to as glucopyranose, whereas this one, it is referred to as the alpha D glucopyranose. So these are the two examples of the pyranose rings which are formed. Now this is the example. This example here is the Howard's projection formula of a 6-membered pyranose ring. But suppose I want to depict this as a 5-membered ring. Alright, if it is depicted by a 5-membered ring, then the cyclic form of glucose is referred to as beta D furanose. Here also we will have alpha D glucofuranose, and here we can write a 5-membered ring, that is furanose ring. But the rule for drawing a Howard projection is the same. This is carbon number 1, 2, 3, 4, 5, and 6. Now, how this 5-membered ring is formed, the furanose ring is formed, is due to the when this aldehyde group in carbon number 1, it attacks the hydroxyl group in carbon number 4. As you can see here, it attacks the carbon number 4. Whereas the carbon number 5, it remains as it is. You can see here this carbon number 5. Alright, so this is a beta D glucose, a beta anomer. Why? Because the OH and the CH2OH, they are on the same side. So the same structure you can show here for the alpha D glucofuranose, where this OH group will be on the opposite side, and everything will be the same. Alright, so furanose rings are formed when the aldehyde group attacks the OH group at carbon number 4 of the glucose chain molecule. The four hexoses here in this example, the glucose, fructose, mannose and galactose, okay, they are all the sugars here. I'm showing an example. They are commonly found in most plant cells. Okay, these are the monosaccharide molecules which are commonly found in most plant cells. Galactose is also found in milk sugars and fructose is found in fruit sugars. So as you can see here, although glucose, fructose, mannose and galactose, they have the same chemical formula, that is C6H12O6. Alright, although they have the same chemical formula, but they differ structurally and chemically, and they are also known as isomers. Okay, they have the same general formula, but same chemical formula, but they differ structurally and chemically and are known as isomers because of the arrangement of different functional groups around the asymmetric carbon atom. Now, all these monosaccharides, they have more than one asymmetric carbon atom. And for asymmetric carbon atom also I had explained in a different video. So as you can see here, all these four molecules, if you take a look at glucose, fructose and mannose and also galactose, they are all hexoses. In case of these first three molecules here, the only difference is in the first and the second carbon atom, as you can see. Now fructose, it differs from glucose and also from mannose in that it is a ketose, okay, whereas these two, they are aldoses. They have an aldehyde functional group, whereas fructose, it has a ketone functional group, so it is a ketose. And the last four carbon atoms, as you can see here, they are identical in all these three compounds, whereas the galactose, it differs from glucose in the position of the hydroxyl group on carbon number 4. You can see here, this is the carbon number 4, this is on the right and this is on the left. That is the difference between galactose and glucose. Now when the hydroxyl group, galactose, we can see that on carbon number 4 the hydroxyl group is on the left. When the hydroxyl group of monosaccharides, example the fructose, reacts with this one, it reacts with its own keto functional group. Okay, the hydroxyl group, it reacts with this keto functional group, then a hemiketal is formed, and for that I had already shown the example of the 5-membered ring fructofuranosis. Alright, so these are the different structural representation of these carbohydrate, of these monosaccharide molecules. Okay, if you want me to show again the fructose molecule, okay, this one I had already showed this before also. It looks like this. You see, when the 6-carbon ketose sugar, that is D-fructose, it forms a ring structure which resembles a 5-membered ring compound, that is furan. Now such cyclic structures, they are termed as furanoses. Thus this one here, since this group, the OH group here, it lies on the opposite with this last carbon atom in a molecule, so this is the alpha anomer. So it will be termed as alpha fructo, that means fructose, and because it is a 5-membered ring, it will be furanosis. Okay, alright, so this is how we should name this, and of course it is a D sugar, so it will be alpha D fructofuranosis. And if the OH group is on the same side with this last carbon atom here, so that will be beta D fructofuranosis. So these are some of the few examples of monosaccharide molecules.